Hello everybody, my name is Asmaa Haja. I am a PhD student at the University of Groningen and my project is part of PERICO, which is a Marie Curie funded project. This work is supervised by Professor Lambert Schoonmaker. I would like to provide you with an overview of my paper, which mainly focuses on the development of a fully automated end-to-end process for segmenting, detecting and classifying of microscopy images of yeast cells. So let's start by defining the problem. Modern microscopy allows the production of big amount of cell sample images in a short time. The large datasets help biologists in studying complex aspects. The challenge, however, is to process the data, extract information, and to identify clinical relevant diseases in a short time. Manual processing might lead to error or fatigues. Hence, integrating techniques from the deep learning field seems to be a promising solution for this problem. The main contribution of this paper is not only to build a fully automated end-to-end process, but also to evaluate a state-of-the-art algorithm for the detection and classification of tiny objects in this field. We evaluated the chosen model on publicly available fluorescence microscopy data of yeast cells images. Each image consists of a bright field channel, which is a grayscale image. The second channel is a fluorescence protein channel, which is a protein that exhibits a specific colored fluorescence when exposed to light. The color green is used here, and this channel is called a GFP channel. The last channel is the merged channel, which is a combination of the bright field and the GFP channels. The library we used contains different protein compartments, called organelles. The image on the right side shows an abstract of cell structure, including the different compartments. The more images we have, the more accurate model we can obtain. This is why we choose images for the organelle that have more than 300 unique images. ER, mitochondria, nucleus, and cytosol are used to evaluate the model. Just a brief explanation of some of the deep learning applications used in this project. Assume we have a bright field image, and we want to do an object segmentation. Object segmentation means that we want to group pixels together that belongs to the same object, in this case a cell. So we can say here is one cell and here is another cell. In the object detection, we want to identify where are the objects. What we do is we surround this object with a bounding box. We can say this is an object and this is another object. Object classification means that we want to categorize these objects. We can say these two objects are cytosol and these two objects are mitochondria. For the object segmentation, we used a mask RCNN algorithm. For the object detection and classification, we used YOLO4 algorithm. As this paper focuses on the application of these models, I will not go into details to explain each algorithm, but I will briefly present the YOLO4 model. In the traditional deep learning approach, each application, such as detection and classification, is considered as one individual stage. Most recently, in 2016, a new algorithm was developed to detect and classify objects by going through the entire image only once, namely in one stage. This algorithm is called YOLO, which stands for You Only Look Once. Its approach makes YOLO a powerful and a fast algorithm. Therefore, we decided to employ the fourth version of YOLO, published in 2020, and assess its performance on the fluorescence microscopy data. For the evaluation of the YOLO model, we decided to define 6 experiments and apply a 5-fold cross-validation for each experiment. In the 5-fold cross-validation, the data is divided into train, validate, and test sets in such a way that each time, one fold is used to test the model, and the remaining folds are used to train and validate the model. The presented table demonstrates the defined experiments. The first 3 experiments uses the original images sizes, while in the last 3 experiments, the original images were divided into 4 quadrants, as shown in the upper right image. We define one-class and multi-class classification problem. In short, the classification of cell images is based on the fluorescence coloring of 1, 2, and 4 cells compartments. The number of training, validating, and testing images are shown in the last 3 columns. The original image will be downsampled to have the same image size as the quadrants. This downsampled image will be used as an input to the model. In the training phase, we first need to merge the bright field and the GFP channels together. In parallel, we segment the bright field channel using the mask RCNN. After this, we create necessary YOLO files based on the segmented image and the merged image. Having all necessary files, we can finally train the model. To test the trained model, we first need to, again, combine the bright field and the GFP channels of the test images I told you about. To emphasize, the model did not see these images before. This is very important. The last step is to evaluate the model based on these images. This is done by various analysis steps, including plotting and providing some metrics to measure the model's performance. Here, the evaluation of YOLO model during training phase is presented. Experiments based on image sizes are shown in the rows, while columns represent the results of the defined classification problems. Although there are more images to train, for the quadrants experiment, the total time needed to train the model is less. This is because there are less cells in these images, and thus the images are less complex. In comparison to full-size images, the detection's performance on quadrant of the images is better. This is represented by the mean average precision measurements. Average loss error computed for the quadrant of the images is around 3.5, which is way lower than for the full-size images. This implies that the classification on quadrant of the images is better than the complete images. Both of these observations suggest that YOLO is able to detect and classify small objects best in native resolution, as opposite to complete but subsampled image. The following table reports the precision, recall, F-IN score, and accuracy on 4 classes for the full-size image on the left and the quadrant of the images on the right. Obviously, the outcome of all these measures show that the classification of individual cells on full-size images is 1% better than on quadrant of the images. The reason for this is because less cells are detected on original image compared to quadrant of the images. This can be seen in the black circle in this figure. The left side of this image shows the detection of individual cells on the original image, while the right side shows the detection on each quadrant of the images. 2 cells are misclassified here and 1 cell here. I will explain the difference between the colors in the next slide. This figure shows the detection and classification of randomly selected cell images based on their fluorescent coloring. Green bounding boxes show a nucleus detection, blue for a cytosol, white for the ER, and red for the mitochondria. Clearly, YOLO 4 demonstrates good detection results on these images. It's also capable of detecting part of cell found on the border of these images. To conclude, we built an end-to-end process that can perform segmentation, detection, and classification tasks for fluorescent microscopy images of these cells. We also evaluated a state-of-the-art algorithm, YOLO 4, on a publicly available data. We showed that by using the trick of dividing the original image into 4 quadrants, the training time decreased and the accuracy increased. For the future, we are planning to make this eScience website available for all interested users. Thank you for your attention.